Miles, I want to take a look at polling question number 2. So we kind of flip polling question number 1 on its ear here, and we're going to ask which of these do you consider the best opportunity to increase R&D efficiency in your organization? So those are your options: new AI, ML, and big data tools, platform approaches to drug discovery and development, adoption of advanced technologies like organ-on-a-chip and in silico modeling, modular integrated high-throughput labs, return of healthy biotech capital markets, growing computational biology talent pool, and digitization of labs. So take a minute and respond there, and we'll take a look at the results here momentarily. Nadim, I want to turn back to you while we're taking that poll question. So Cullinan's discovery process is anchored, as you said, in target first, modality second. How is, like, what's that early decision making around targets to explore specifically? And then what is the thinking behind then applying the right modality and mechanism of action to that target?
Sure. Yeah. Look, the good thing is that members of our R&D team, scientists, clinicians, and clinical research scientists are very closely connected to kind of emerging research and the most promising oncology targets out there. We're very well embedded within the broader scientific community. So again, it's having that internal and external ecosystem that you can tap into, which is really important for us. So that's one thing. And then I would say, as well as being modality agnostic, as I said earlier, we're also agnostic to the sources of innovation. Again, whether it's internal or external, I think that we have the humility to understand that there's great thinking outside of our building in one main street, and we deliberately target that so that we can tap into that scientific innovation. And basically, we do the best innovation. It doesn't matter where it comes from. And I think our teams are really good at being completely unemotional to whether it's internally developed or externally sourced when it comes to advancing our programs, which is really good and very, very important. And we've successfully sourced innovation from academia, from industry, for our internal efforts. And then we can apply our own preclinical and translational research expertise to then advance those programs. So I'll give you one live example of how we've done that. So, we recognize that MIC-A and MIC-B could be a very important target in cancer. So MIC-A and MIC-B are ligands for the NKG2D receptor found on NK cells, subsets of T cells. And what happens in cancer patients, the tumor cells can shed MIC-A and MIC-B because of proteases cleave MIC-A and MIC-B off the cell surface in the tumor microenvironment. So it once again makes those tumor cells invisible to the immune system. And so we thought about, again, with this target mentality approach, how can we restore that MIC-A, MIC-B, and NKG2D axis so that the immune system can once again recognize those cancer cells and destroy those cancer cells? So that led us to in-license a molecule called CLN-619, which is a humanized IgG1 monoclonal antibody, which works through many modes of action, including binding to and stabilizing MIC-A and MIC-B on the cell surface and other mechanisms of action, which again make those tumor cells visible to the immune system so they can't evade the immune system. And so this was a really important program for us. And, we did the preclinical experiments, we licensed it in, we took it into the clinic. And so we knew there were multiple mechanisms with this molecule, but we took a target and then we identified an asset with the right modality to address that target. And then we designed a program around it. Again, way before anybody was paying attention to MIC-A and MIC-B. So we currently have the only MIC-A, MIC-B antibody in the clinic. And fortunately, last year at the American Society of Clinical Oncology meeting, we shared our initial monotherapy phase 1 dose escalation data. And we were able to see single agent efficacy across a range of tumor types, which is quite unusual in oncology in a phase 1 dose escalation study. And it included patients whose disease had progressed on prior checkpoint inhibitor therapy, an important clinical problem we were addressing. So this year now, we're looking forward to presenting the first combination therapy data from that program in the second quarter of 2024. So that's just an example. And so as we apply that same thinking broadly, it's given us the result of a portfolio of very distinct potential first-in-class and best-in-class programs. So again, this was that taking the target approach first, and then finding the best modality to address that target, which yielded a first-in-class program, which is the first in the clinic, for which we think is a very important target.
Yeah, you earlier talked about the CD19 program being you know, not first in class but a best in class approach. It occurs to me that this one is it's first in class, I guess, by virtue of its first in class status. It's also best in class, at least for now.
That's right, exactly. Yeah, when we can get, no, I'm a commercial guy. When you can get first in class and best in class, you know, obviously that's great. But either of the two are equally valuable.
Yeah. Very good. Well, I appreciate those real world examples. We're going to pull the polling question results up here real quick and reflect on those for a minute. Miles, if you can. All right. So new AI, ML, and big data tools looks to be leading the charge. Any surprise there? I mean, Bob, that can't surprise you.
No, you know, it really is where the industry has been the past few years, looking at small molecules and then biologics, but then really where we're going to get to in terms of understanding the patients in more depth and the disease states. It is the future of where we're going to get to.
Yeah.
Yeah, I agree, Matt. And I think also, in addition to addressing the big problems, I think there are some of the, I guess, less transformative but equally important opportunities, you know, when it comes to things like even, you know, regulatory document writing, medical writing, that you can use some of these large language models to really make that process much, much more efficient than it is today.
Yeah, that was another resounding theme. So, you know, Nadim, I know you were at JP Morgan last week, and Bob, you didn't make the trip out there this year?
No, I was not there this year.
Yeah, you're the lucky one. The conversations around AI and ML, as I said, they were pervasive, but a lot of the conversation was what you just said, Nadim. Like, what are the applications that we can look at down the road in clinic where, you know, where the money really starts being spent in a big way? How can we apply those efficiencies down the pike? So we're running short on time today, so we'll put that one on the back burner for a future discussion on Bioprocess Online Live. But I want to give you each an opportunity to wrap up. I know we didn't get through our entire agenda, but maybe just share some. Bob, I'm going to start with you since you're large on my screen. Maybe share some sort of vision for where you plan to drive R&D and R&D efficiencies at Amgen, especially sort of with you know, the new launch point that you're at after the acquisition fourth quarter last year.
Yeah, I mean, the point that really excites me moving from a smaller pharma company like Horizon into Amgen is this access to the technology scope that is pretty amazing within Amgen. They've built a lot already in terms of looking at artificial intelligence and machine learning, and that really has applications for the drugs that are moving forward. And you know, the next thing that we're really looking at there is to, you know, try to understand how we can get towards, you know, better therapies in patients. And then this undiscovered like space essentially where we can actually get to things that are more undruggable or more difficult to drug, and really try to tap into that. So for me, that's really where I think the future lies, and very excited about you know, the future within Amgen and within the industry in general, about the things that we're going to get to and really the benefit that it's going to bring to patients.
Yeah. Nadim, parting thoughts on the vision for R&D efficiencies at Cullinan?
Sure. Yeah. For me, one of the key questions there is, for us, efficiency means being clear on what we're focusing on, but also on what we're not focusing on, which is just as important often. I'll give you an example. So for us, today at least, complex modalities such as gene and cell therapy are currently off strategy for us for a whole host of reasons. And I know that space very well, as you know. Secondly, I think continuing to have this high bar for go, no-go decisions applies for me for both discovery and development. So if, for example, a program in the clinic doesn't clear our go, no-go criteria for phase 1, we're not going to take it into phase 2. And we're going to move our teams on to other programs and reallocate our resources. If we have multiple programs, of course, that pass our go criteria and generate clinical data, well, that opens up a lot of value and strategic optionality for us as a company, including the opportunity to have, potential external partnership opportunities. And then the last thing I'd say, look, as a company, we deliberately chose that our mission is to create new standards of care. So that means our progress is not planned to be incremental. Look, it's important to get FDA approval and clinical acceptance, but I think we need to do better. And I think by continuing to strive for therapies that produce, highly durable outcomes, more tolerable experiences for patients, this kind of modality agnostic approach for us, it gives our teams incredible freedom to discover and develop a range of diverse cancer therapies that hopefully will fundamentally transform outcomes for people with cancer.
Excellent. Well, guys, I appreciate you both for coming on and spending some time with us and sharing your insight, and for staying on for a few extra minutes here. I apologize that we went over. I didn't quite get to everything, but like I said, there's always room for a part two. But thank you, Nadim and Rob. I really, really appreciate it. Miles, let's give them a virtual round of applause. You got that? You got to hit the... No round of applause. All right. Virtual round of applause for Nadim and Bob. And thank you. Oh, there it is. There it is. Yeah, a little late. Didn't have that queued up. But thank you for supporting the program that we put on, the greater industry. Really appreciates your expertise. I want to remind our audience that this discussion will be available within 24 hours to view on demand at bioprocessonline.com. Please share it with your friends and colleagues. You can use the same link that you used to register. Stay tuned for our next program on March 19th at 11 a.m. when we're going to tackle next generation manufacturing analytics and control technologies. In the meantime, subscribe to the Business of Biotech podcast. Visit the full archive of Bioprocess Online live events at Bioprocess Online under the live events, under the Listen and Watch tab at bioprocessonline.com. And we'll see you next time.